Hello everyone, my name is Alonso. I'm from the Scientific School Research Center from Santiago de Chile.
Hello, my name is Tyto. I'm also from Santiago, Chile, and I will introduce you to a project titled Detection of Potential Hantavirus Reservoirs through the study of pellets from lesser hornole and barnole. First, I would like to tell you more about the hantavirus. It is triggered by a retrovirus with the same name, and it has two main strains, cell variant and Andes variant. The confirmation of hantavirus in Chile happened in 1995 in the South Zone. It is classified as a zoonotic disease, meaning that the way of contagion goes from animals to humans, in this case from rodents, via shed particles in the air coming from feces, urine, and saliva. In Chile, we have 7 species of rodent that are classified as hantavirus vectors. From the 7, I would like to highlight the last 3, the long-tailed mouse, the main vector, and the 2 latter, the black rat and the house mouse, being both alien species in the territory. I will also tell you about the SNASPE, protected areas. In them, you can find different species of raptor where these two stand out, the barnole in the left photo and the lesser hornole in the right. Both are natural controllers of the rodent that transmit the hantavirus to the human population, as a research made by us in the year 2017 showed.
Here in this map, we have the geographical distribution of some of the pathogenic hantavirus strains. First of all, we have the Hantavirus cardiopulmonary syndrome that is located in America. This is also called New World hantavirus. We also have the hemorrhagic fever with renal syndrome that is located in Europe and in Asia also. This is called also Old World hantavirus. But the most important thing that you have to keep in your mind about this map is that the hantavirus is a public health problem all over the world. Here we also have some clinical features about the hantavirus. From day 0 to 10, after the infection, we have the incubation period, and then from day 5 starts an increasing of the IgG and IgM antibodies. And also, some of the symptoms of the first step of the disease are very similar to the flu, like myalgia, bladdache, visual disorders, headache, and in the last step of the disease, we have shock hemorrhages and renal failure, ending in death in the 25 to 40% of the cases.
A problem that we face in our country is the way to identify the vector rodents in the protected areas. It is carried out by trapping alive individuals, exposing those who manipulate the traps to a high risk of contagion by hantavirus. And not only them, but the thousands of tourists that visit the protected areas throughout the year have in direct contact with the local fauna and therefore with zoonotic disease vectors. Another problem that we are having is that the public health services describe the presence and representativeness of the main vector Oligoryzomys longicaudatus just by region, not specifying its presence inside the protected areas. This rodent has the highest seropositivity for Hanta. This is the probability of finding an alive infected individual. So this is a must that we have to do as a country, and not only for the long-tailed mouse, but for every vector rodent.
Because of that information, we developed the following research question. What is the representativity of rodents that behave as potential hantavirus reservoirs in the system of protected areas in the north, center, and south of Chile? We also developed the following hypothesis to this question, that is, due to the high register of hantavirus in the south of Chile compared to the rest of the territory, it is expected that the representativity of potential hantavirus reservoir rodents found in the diet of the raptors would be greater in the areas of the SNASPE located in the south in comparison with the rest of the territory. And to get accomplished the research question, we developed the following overall objective, that is, determine the representativeness of rodents that may behave as reservoirs of hantavirus found in the pellets of the raptors that inhabit natural protected areas in the north, center, and south of Chile, in order to establish potential risk zones of transmission of the Hantavirus pulmonary disease for the population that lives nearby the protected areas.
Then we broke this up into 3 specific objectives. The first one, to quantify the individuals of each identified species of rodent that were prey on by the raptors that inhabit the areas under study. Then to determine and quantify the dietary items of the raptors that may behave as potential hantavirus reservoirs. And finally, to establish the zones that could present a high risk of contagion by hantavirus. In order to carry out this project, we separated the methodology in 3 parts. The first, the fieldwork. Between the years of 2018 and 2020, we did 12 field trips going to 6 different protected areas, 2 in the north, 2 in the center, and 2 in the south. They are all very different ecosystems. Then, in the map, we can see how are these areas distributed. In the north, we visited the Fray, Jorge, and Las Chinchillas National Park and Reserve. Then in the middle zone, we went to Rio Clarillo and Peñuelas National Reserves, while in the south, we took samples from Villarrica and Conguillío National Parks. Once in there, we identified the perches of the raptors and proceeded to collect the pellets that we could find under.
Here we have two pictures of pellets. Pellet is just the digested material that the raptor vomits after he eats a rodent, for example. Inside, we can find the skulls and the hair of the rodent that was preyed on by the raptor. This is extremely useful because I can identify the species of rodent that was preyed on just looking to the dental morphology. I can compare it to the other keys that I will explain next, but the main difference between these two pellets are that in the hornal pellet at the right, I can find some shiny residues of covering. In the barnal pellet at the left, I don't find that.
In this video, you can see my mate under a perch in the Rio Clarillo National Reserve. A perch is the place where the raptor stays, sleeps, and regurgitates this bulk on the digested material. Identifying these places is not that easy, but once we could, we picked the pellets from lying over the surface, built on the perches, and carried them to the laboratory for the lab work.
For the lab work, the collected pellets were immersed in oxygen peroxide. This was to facilitate the separation of the skeletal remains, and we took the skulls of the rodents that were preyed on, and these are the keys that I told you before. At the left, you can see the dental morphology of that rodent. It's the same that we found in the skull. So we identify at level of species.
Finally, the rodents obtained were quantified and associated to potential hantavirus reservoirs. We elaborated graphs and tables in order to subject the data to a statistical analysis. Here, you can see your very first results of the rodents found in the pellets distributed by geographical area. In orange color are highlighted those species that are described as vectors of hantavirus. In the north, we found a lot of Phyllotis darwini and one individual of Abriotic solivaceus. In the center, we have a more uniform distribution, finding 5 different reservoir species, but the importance here is that the Oligoryzomys longicaudatus, the main vector, is appearing in our samples. Finally, in the south, we found only vector rodents and undetermined basseriforms, but we would like to emphasize the big amount of Oligoryzomys longicaudatus here. Here, you can see a graph that shows the representativity of reservoirs in red color, again non-reservoirs ones in blue. As you can see, as we move to the south, the representativity of reservoir species rises, going from 53% in the north to a 95% in the south. Now, if we just focus in the representativity of Oligoryzomys longicaudatus, we see 0 individuals in the north, 21% of representativeness in the center, and an impressive 55% of representativity in the south. Is it important to focus on these species? This has seropositivity that surpasses the 7% in the south zone.
For the statistical analysis, we create 2 questions. The first one was focused on to know if the composition of the diet of both raptors between the areas was different. So we carried out a non-parametric multivariate analysis of variance with a 99 level of confidence, and we obtained a p-value of 0.003. So this indicates that there are significant difference between the diets. For the second question, we wanted to know if there was a significant difference, but now in the representativeness of potential rodent reservoirs between the areas. So when we compare north and south, we obtain significant difference. When we compare north and center, we also obtain significant difference. But when we compare center and south, we didn't obtain significant difference. This is really interesting because if all the cases of hantavirus are located in the south of Chile, why I didn't find significant difference between center and south? We're gonna discuss next.
What you have to know is that the south of the country presents favorable conditions either for Oligoryzomys longicaudatus and for the virus to stay out of the host. The south has a very high density of Quila, a plant that might be used by the rodent as refugee or as a food source. The temperature, the short time of light exposure, humidity, and the big number of human settlements allows the virus to stay out of the vector for up to 13 minutes. So we can infer that the high number of cases in the south is due to the high representativity of the main vector, its high seropositivity in the south, and the favorable conditions for the hantavirus and its vector. In the north and the center, it is totally the opposite. The main vector is not that represented, its seropositivity in those zones are lower, and the conditions are not favorable, and there are presented less human settlements.
The areas with the highest potential risk of contagious are Villarrica National Park and Conguillío National Park, because these were the areas with the highest number of vector rodents. Also, it's necessary to extend some campaigns of the Ministry of Health in order to reduce the risk of contagions in the parks. Also, this method is proposed to study hantavirus vector rodents, but now I reduce the risk of contagious because I don't trap the rodent directly. Also, the just identification of the potential rodent reservoir is not a complete work because I have to define a risk zone. So for this, it's necessary to perform biochemical methods to the pellet to know the rodent that was parthenite had or not hantavirus.
So in conclusion, the representativeness of potential hantavirus reservoirs is higher in the south and therefore a higher potential risk of hantavirus infection, mainly due to the high presence of Oligoryzomys longicaudatus. It is added that the use of pellets of generally feeding raptor is a reliable method for the identification of potential hantavirus reservoirs, fulfilling the objectives and upsetting the working hypothesis.
Also, some of our projections are, first of all, to expand the areas under study covering a larger geographical zone. Also, and this is the most important projection that we have, to explain to tourists the results of our research for them to know what they are facing when they visit these parks. The hantavirus is extremely lethal disease, so they have to know. Also to evaluate a biochemical method to determine if the rodent that was part of the diet had or not had hantavirus. These are some of the references that we use to carry out the work. Here we have the QR code, and you can scan it to go to the full project. You can pause the video to go there if you want more information. And these are the institutions that we work with. Thank you all for your attention. Hope you have liked it.
Thank you for your time.